2-1. Okay, here we go.
Yet another evolution hour, continuing in our happy little thing all through 2020. We'll see what happens on that. We have a guest as well, which Jackson will be able to introduce since he's aware of him, a botanist, and so we'll be kind of veering off stream to discuss a lot of subjects, and who knows where it'll all go. For those who have been following the show for the last umpteen forever, I've been going after this Contested Bones book. Somebody had called attention to it in one of my internet discussions, and they said, would you want to do a review of this thing, because the idea of my having to read it is repellent? And so they bought it for me and sent it to me, and I've been exactly doing that, because the whole issue that I go into is source methods. If you really want to see if they're behaving reasonably or not, take a look at the references, look underneath at the source data and how they're playing fair or not with the source material. And that's a dead giveaway as to whether you're dealing with pseudoscience or not. And boy, are we dealing with pseudoscience here. Rupi and Sanford are young Earth creationists, although Rupi is kind of vague. He doesn't officially deal on that, but the fact that he gets published in creationist journals is kind of the clincher on that. But good old John Sanford is one of our little bet in the wars that I have to in a critics background, helped invent this gene gun thing that's used in some biological activities. Comes from, I think, a plant background. You might have heard of him from that direction. But he's dedicated to a young Earth creationist model that he doesn't always tell people about, but he really wants to try to show that there's been a genetic entropy deterioration of all the organisms in the genome since all 6,000 years ago and the 500 years ago. Needless to say, he's had a problem documenting that. And it's just absolutely fascinating as to looking into how he's misrepresented primary sources and evaded things. And I've been going through the book, although Sanford is actually kind of the caboose in this work, because he freely admits that Rupi is the one who actually did most of the research, quotation marks, because an awful lot of it is simply siphoning off other creationist apologetics. He's not actually an original one. In my Troubles in Paradise methodology of creationism work, I differentiate between merely being an apologist, even a prolific one, and being a fact claimant. In other words, how many people actually are the ones who are making the claim, and on what basis, as opposed to the larger group that are copying the claim and spreading it out. And it turns out there's only about 60 fact claimants in all of anti-evolutionism, and we pick off an awful lot of them in the Rocks Were There project. Anyway, the current bit that I've got to, Lisa says we're cutting in and out.
Well, I've been monitoring it. It's okay for the most part. We're doing all right now.
I've got my files cut down to a minimum here, so to keep the data stream down, and so it's just up to the internet gods. We apologize profusely to those who, if we cannot be heard clearly, we'll wing it, as the case may be. Anyway, the thing where Sanford and Rupi got my dander up on this particular page is they took on the Dover Kitzmiller trial, and they just alluded to it in a pot shot where they claim that it was a miscarriage of justice. Because at one point, Ken Miller had brought up a particular pseudogene, and it turns out a little bit after that, there's elements of that are under selection pressure. Even though the article, I put the linkage up to the article. One of the things, Obuzgo, that I like to do in my work on all the video material, and Jackson does the same things in spades, is we provide the direct linkages to the works in our video description. So nobody has to take our word for it. They can, and in fact, when I've been watching videos, even very good videos where they don't really put the direct sources up, I say, bad dog, you should actually put linkages up, at least to core critical material. And it needs to be to full text material. So although I do encounter an awful lot of ones that are available in abstract, if a general person out in the real world can't get the full bloody text, I don't put the linkages up for it so that they can actually follow up and read the whole thing. Anyway, this particular paper that I put the linkage up on, Svensson from 2006, starts off by pointing out how most of the pseudogenes are in fact pseudogenes and don't do a damn thing. And, and so they were basically stepping, hasn't changed a lot. But anyway, the Dover trial, of all the things to call a miscarriage of justice, the Dover Kitzmiller trial, which for those who aren't familiar with it, from 2004 to 2005, this is where a group of wacky ideologues, including ones who were following Kent Hovind, wanted to get of pandas and people the semi-intelligent design. Yeah,
I think
you've heard
Buzzgo
of
knows
that
about
one,
this.
yes.
Yeah.
The pandas and people textbook into the Dover school system, and they did it by stealth. And gradually, some parents started to say, whoa, wait a minute here. So this supplemental text turned into a court case, and a bunch of people in the intelligent design side were going to be coming in to testify on it. And then they started peeling away as it became painfully obvious that there was going to be a big problem here. So pretty soon Steve Mai there and Bill Dembski's not going to be there. And it just turns into a debacle for the intelligent design side. Both sides, ironically, wanted the judge, Judge Jones, to rule explicitly on whether intelligent design was science. So, surprise, he did that. And he said, no, it ain't. And he relied on an awful lot of things. And just from a technical point of view, one of the things that was particularly scurrilous is that Burlington, who was on, I think, opioid addiction, the guy who was the conservative who was trying to get all these books in there, he lied under oath to the judge about where these books had come from and who had paid for them and the circumstances of it. And lying under oath thing is kind of a serious point in a district court judge, so basically this was like a foregone conclusion. Anyway, intelligent designers have been screaming bloody murder all about it ever since. Casey Luskin had gone ballistic over and over and over again on it, and I put the linkage up to the Mirror website, the HTML version of tip 1.7, where I go into the Dover trial and the grisly details of what the hell was going on in the reaction. You can also find it at my main website in a full PDF. Yeah, jump in there, Jackson.
Buzzgo, is there anything you'd like to add?
When I came from Florida and originally Europe to Louisiana, Shreveport, which is just a few miles away from the Bible Belt, and I did have students out on me when I used the E-word.
Ooh.
The E-word. I had students walking out on me, and I changed the coach. Now, based on Popper, that's a book you probably know. Mm-hmm. It's available in every bookstore or online. Karl Popper, he has a very nice definition of science and knowledge, predictive power. The key part of predictive power has a very clear classification: money. You can predict the weather and you're a farmer, or when you're in stock market and you want to predict what the stocks are going to do tomorrow, like in a margin call, when you know what's going to play on Wall Street tomorrow and make millions or lose them. And so predictive power is everything. And if you have a model that can predict what you're going to find, either in terms of ecology or in medicine, that is better than another model, take it. So we have two systems. One is evolution, and the other one is the little book of the green gnomes. Each one is going to give you better prediction about resistance of diseases, development of new diseases, medication, or what you're going to find in organisms in terms of ecological interaction. Book of Gnomes or evolution. If Book of Gnomes is better, go with it. Otherwise, stick with evolution.
Oh, Jackson, because that's several people in the audience in the live chat, introduce our guests so we know who we're talking to.
Yes, this is Dr. Buzzgo. I'm sure he can introduce himself. Tell us a little bit about yourself.
Well, you introduced me as a botanist. I did I earned my spurs and my degrees on botany topics, but in evolutionary context. So the beginning was basically the evolution of early flowering plants, and particularly monocots, so things that have only one cotyledon. And I worked intensely with sweet flower Acrocalamus, which is the sister to all monocots. And there I discovered that morphology was not sufficient to describe the homology of these organs that these plants have. It's this kind of, what the heck, what is it? Is it a petal or is it a tract? Both. It has both features. And I described to my professor, and he was completely confused, didn't like it very much. And then I discovered works from David Bohm, Meyerowitz, and Enrique Cohen, The War of the Worlds, one of the famous papers in Nature, where it goes about different gene categories that regulate flower development. And there's 3 classes, the ABC classes, and they basically contain the answer to my question of my PhD thesis. Then I became an evolutionary developmental scientist and worked on a gene expression pattern, particularly in the University of Florida, where we studied those genes that make flowers look like flowers in ancient. And that's basically where I come from. So a big part of what we do is try to understand or even predict what a gene is going to do. So if you have a gene like Apetala 3, or if a gene like Pistillata, and we say, if we knock that gene out, this flower will not have petals anymore. And then we take what's called a orthologous gene, so basically the same gene sequence from another plant, and put that in, and then it has petals again. So imagine yourself, you have a little Arabidopsis, the false tail crest, and you knock out the gene that makes petals, and it's an ugly plant to start with, but it's ugly. It doesn't have any flowers. It's just green. Or the flowers have no petals. They're just green. And now we take the same gene from tomato and put that back in, there's petals again. That's called rescue. If you think that global climate change ain't happening, and why don't you go and buy land, real estate down in New Orleans right now? I'm sure you can buy a lot of land at sea level at New Orleans. Global change won't happen. Global climate change won't happening. Go ahead and buy. Go down, and
the Evo Devo crowd over with birds and mammals and others are starting to do paleogenomic reconstructions to where they're tinkering around with ancient genes to figure out what the system was doing before it reached the stage we have now. And so they're recreating what the archosaur snout of birds are. They're working out the tooth structure of mammal teeth going back 100 million years and all that. Has there been much paleogenomics work in trying to reconstruct these unusual forms that are found like Cretaceous angiosperms or even farther back in the plant crowd?
Well, the fun thing is that you actually don't find much further back. That's the Darwinian abominable mystery. Just somewhere, the oldest fossils that I know about are one is Archifructus that looks, I'm not sure even what's called flower is a flower. It could be an inflorescence with used flowers on it. And the other one, Leafructus, which is a delphinium or columbine, which is in the eudicots. So it's a crown group of flowering plants, which is the oldest whole fossil that we have. It's like
those darn plants should get fossilized better.
Well, the thing is, yeah, indeed. And so basically, it's the same situation if you ask about the origin of the telephone, and you find Tutankhamun holding a cell phone in his hands. Explain where the phones come from. So this situation is what we have, and so we don't really know what we have to develop, but we have a bit of clue exactly from this ABC model that plants around Ambarella, Cabomba, and star anise are old models, and so it's a bit hard to move plants that far back because we had a big shift in the flower genomics at the base of the core eudicots. Actually, basically, the petal got lost and got reinvented, similar to the snakes of eyes. I'll be the same situation here. And to bridge that backwards is really difficult. So we can't basically make a rose totally look like an ambarella, but we can show how these transitions happen by a multiplication of genes in the eudicots that allow them a more sophisticated regulation and a more clear, distinct regulation, while the ancient angiosperms don't have these clear distinctions. They have gradual transitions from bracts into variant organs. Dangerous to call them petals and sepals at that point. They transition and then even transition into stamens. So this transition was shown in Ambarella, and this has been known for a long time also for magnolias, and that actually more articulates with genes. There we can play around. That we can do. We can mess around with the rose, changing its stamens, the male parts, the pollen-producing parts into petals. We do this in every shop. When you buy a rose on Valentine's Day, you wild type rose just falls apart after 2 days. you buy these full heart-shaped roses from Colombia, these beautiful love roses, which have many petals. How did that happen? Well, the stamens on the periphery of the androecium, they basically got castrated. They lost those genes that make them stamens and lost their stamen, became their
You mean people
petals.
are giving out their anniversary presents of castrated flowers? I'll tell you.
Yes, partially
It's
castrated
disgusting.
flowers is the sign of love. Does this tell you about Valentine's Day? So
Yikes.
roses, double roses, filled roses, or roses where the expression of the agamous gene has retracted from the outer androecium to the inner part, or completely lost, depending what breed you have, how full it is, and convert then the stamens to the staminodium.
Is it also an intimidating factor that compared to us boring mammals, plants actually have a hell of a lot of genes?
And they can do more things with it. In a mammal, when you go mess around with the genes very quickly, the animal just dies. So if you take off your Hox genes away, the easiest part that you get is that you have a spina bifida. You can survive that. But if you mess up the ending of the head and the butt, that's not a good life anymore. You're not going to get very far with that. And so that's what very quickly happens. The same with Drosophila. If you mess up the Hox genes, then not much is going to happen with that little Drosophila fly. It's not going to develop very far. And in flowers, you can mess around with those genes a bit better, and you still get something that you might be able to reproduce out of a tissue culture, or even with some rescue breeding experiments. You can rescue the plant and bring it through. With animals, it's not that easy.
There was a paper.
It's better.
There was a paper that came out at the end of last year, which was looking at Marcantia and talking about how I believe it was the leaves. I think it was the leaves were like independently developed.
The thallus.
Oh right, it said they were
Oh,
independently
yeah.
developed, but from the same set of genes, wasn't it?
Yes, in all these so-called cryptogams, the non-seed, non-vascular, basically pretty much the non-seed plants part. The non-seed plants still have a gametophyte in Mercantia. The green thing is the haploid gametophyte, and also then in fern on the other side, the gametophyte is very small. What you see as a fern plant is the sporophyte, but that interchange is actually possible whether you're haploid or diploid. So they could make gametophytes become sporophytes, and they could make sporophytes behave, or diploid tissue become a gametophyte. There's also an experiment of Fiscomitrella that this happens, and that's actually part of the things I wanted to do, but I got spooned. I couldn't produce that kind of science. I was interested in that, and I follow this. There's a lot of work done by Joe Bowman, and also people in Florida, and also by Ned Friedman. He has also been involved in that.
You might get a kick out of one of the topics that we address in the new Rocks Were There book. Fasten your seatbelt on this one. That according to Steve Austin and Kurt Wise, coal forests came about because there were gigantic floating forests and ecosystems pre-flood that then got swept in post-flood and baked into coal seams in the course of maybe 5, 10, 20 years.
I love the look on his face.
Needless to say, it's an idea that's cranial blockage of the rectum stupid.
In that sense, I'm sure we find huge coal layers somewhere in the deep sea ocean when these big floating forests sunk and ended up as huge coal layers somewhere in the Mariana Trench. I'm inviting this gentleman to throw up some $1 billion for an exploration of the deep sea ocean for coal.
Oh, and by the way, we've also got a section on the wacky oil industry, or rather the creationist oil industry, because there's been a small number of, let's be charitable and describe them as loony hucksters, who have tried to use biblical creationism to locate oil in Israel.
Go ahead. It's brilliant. That's what I say. That's brilliant. That's pretty. If the book of Josiah helps you to find oil layers where those stupid scientists are completely clueless about, go away. That's to do. I mean,
T.D. Lane in the live field calls it easy baked coal.
Yes. I mean, this is what the market calls disruptive technology, right?
Mm-hmm.
I mean, this is the unforeseeable foreseeable, like the black
Yeah.
swan.
Put your money down, give it a whack, and see what happens on it.
Yeah, go ahead and invest your money on biblical and Genesis-based oil exploration. Why not? I mean, there's a lot of investment models now that are based on religious background. There's the system that's based on Sharia and Islamic concepts of investments, and it works. It's not like this is all bogus. So why not a creationist investment fund? Strictly invest.
Well, back in the day, Sheikh Yamani,
the Bible. Why not?
Sheikh Yamani, the head of the oil OPEC cartel, used Arabic astrology for making decisions, and it worked for a while, but
Oh, this is
not
very
indefinitely. We're almost near a half hour. Let me go ahead and thank my patrons here, which I do usually in the middle of the show, and I think we'll have probably a massive amount of discussion after that. So I want to kind of get that little thing out of the way. Everybody that's been helping the project over the years, it's made life a little bit easier for old Social Security RJ here. We've had our colleagues Hendril and Eric and Speed of Sound and Surus, and our researcher level Travis and Convert and Eat and James and Ralph McFadden and Pelagia and Benjamin Simpson and Ugly German Truths, and our assistant researchers Mike and Ian and Duranko and Totis Real, and our friends Daniel and Steve Bauman, who is reading the geology chapter. Marigale Beddoes, Insects Are Cool, Devin Miranda Reeves, Morton Nielsen, Puffalophagus, Bo, Staggles, Alex and Paul, and then legacy patrons who were able to help at one time or another, Jen and Jody and John and Keith and Andrew and Dyer and Yui and Mona and Brad and Daniel and Nyanya and Sun, Sky, Stone, and Everett, and Tour, and Zeshi. Thank you all for helping out all of the time. It's made life easier. There's a linkage to the Patreon account and to my GoFundMe at the video thing. Help out as you can. And that gets that little thing out of the way. The second part of the show, I was going to have a thing. I like to have kind of a two-party thing. And one of the bits that
RJ, I have interesting news, though. Yes. So, you know how we usually get, like, an infinite amount of time to do a Zoom?
Remaining meeting time, 8 minutes.
Yeah, so we can start another one once it.
Yeah, we'll proceed under the assumption that 8 minutes is real, and we're about to be unplugged. So
it's going to kick us. So
yeah. I'll be very quickly pointing out what the hope was with the gene code. We're discussing the amino acid associations and whether or not that's actually information or not, and it's not information in the way the creationists want to. And then I thought, well, shouldn't we have like a chart of the amino acids? And then I thought, well, that actually is interesting because some of the modern views are that the amino acid code may not have involved triplets initially, and that some of the amino acids are more easily synthesized and were the earlier ones in a prebiotic, not quite genetic code. And so I thought maybe that would work for a footnote, which would become an info box. And I thought, there's actually a hell of a lot of new information on this subject. So it becomes an appendix. And I put a linkage up to the paper by Catano and Olis from 2016, which goes into an awful lot of work on what the amino acid system was. And this surprised me when I read it. The idea that the amino acid nucleotide associations don't necessarily start with the code. In other words, the system with the ribosomes is a late player that uses the associations for nucleotides because that's what the molecules were doing anyway. So that's where the coding system comes along. And surprisingly, the little wimpy proline of all of the amino acids is very probably the very first amino acid that linked up stereochemically with the C codon, the nucleotide.
I think that was part of, I think Longo and the others figured that was part of the prebiotic set.
Yeah. And so I've been pulling together some information on that. Plus the fact that there are three interesting areas that are involved. One is this idea that there was a precursor code that involved fewer codons, that the wobbly third codon is producing a lot of the variation. That's one element. Second one is that various organisms over time have added on changes in the system. So one of them has converted a stop codon into a tryptophan code. So it now has two codons clusters for tryptophan instead of one that everybody else does. And all of those they can even see in transit to where they're in the course of changing a codon association in some of these little mitochondria. That's another fascinating issue. And by the way, Paul Nelson, the young earth creationist, air quotes, intelligent design guy, put his foot down, stamping on the fact that because there are variations on the genetic code, therefore there is no such thing as a universal genetic code. And he just didn't bother to look close enough at the details to realize that we can tell what's happening. But Buzzgo, you're about ready to spit and nail, I bet. Yeah. And then the other fascinating with this codon reassignment is that they can see it in practice. So what I thought was there was actually an awful lot of information there that is relevant to the origin of life question, kind of coming at it from the back door to this prebiotic system where there were these gradually expanding associations. There are amino acids that are almost ubiquitous in origin of life because they come up so easily, they're easily generated. And it turns out that in just adding on a third codon, but it's still in a duplex code, still you can pick up every one of those amino acids. And so it's looking like the core training wheel set. Well, the other aspect is the fact that there's two extra amino acids. There's selenocysteine, and then the one whose name I keep on stumbling over, pyrrolene, blah, blah, blah. It's got too many letters in it. That's the little pyrrolene used in archaebacteria, archaea and bacteria. And it turns out those Loki bacteria that they just discovered have kind of an intermediate mode when it comes to that particular one. And so there's some various work that Cavalier Smith. Again, there's interesting little tidbits. I'd actually.
I'm just like champing at the bit for new papers on the Loki archaeota.
It's going to be one where anybody that wants to keep their tabs on something that's going to be really interesting research will be that bunch, because it's odd to classify precisely because it's a little bit of this and it's almost that and it's not quite this. And
so what are your thoughts on that, Buzzgo?
I haven't heard about Loki bacteria myself or Loki archaeans yet. But I think a lot of this part in the archaeans, they're understudied because they're hard to get. They're not well studied yet. They were actually discovered or described while I was a student, so they were quite new. Yeah. And there is a lot of unknown part. For me, very interesting is much more where basically the archaeans and bacteria meet, because they do have similarities. They have the same type of nucleotides and stuff. So there must be a common denominator. And how good from each other. Very often when I look at bacteria, they have some advanced things like the double membrane. I don't know where that comes from. That things. The other part is, of course, the beginning of cytoskeletons, how the entire sexuality evolved. I think the further back we go, the more we lose parts because they have died out or have not been discovered yet. They might live somewhere, and there's just too much wide territory to really walk out on the thin ice of speculation.
Based on just living organisms. That just as we know, so many animals have died out over time, and there's so many missing things, and yet we know just from the fossil record there are animals that will have hundreds of millions of years of runs, successful trilobites, ammonites. They had their state, and they're totally extinct now. How many of these bacterial forms represented very dynamic systems that had an impact on the biology of their time that we can't look at now because they didn't make it past the Ediacara and mass extinction?
Right. And of course, one of the big hopes that I have is that we find basically the platypus of archaeans and the volapine of the bacteria. That we find living fossils, because fossil archaeans and fossil bacteria won't tell us very much, especially not come like 2 billion years ago. It's just going to be a carbon smut and we don't see anything in it. But if we find some surviving organisms, living fossils, so to speak, in areas we haven't looked at or areas where we haven't found them yet. I just think about some of the fungi relatives that were found in school ponds outside of when they made the environmental PCR, and went out to the school pond, got some pond scum up and screened that and found a new kingdom, or basically a new branch between the fungi and the animals.
Below a minute, we're going to disappear shortly.
So what I'm thinking is
That's what I think we hope for.
Yeah, definitely. So what I'm thinking is gonna have
Or
to happen
finally.
is we're gonna cut the stream, or we're gonna have to cut the stream. But I can email the new links out. I'll just basically it'll just kick
And
us
we
in
can
the,
restart. So stay tuned,
yeah.
folks. I'll set up a show for a secondary thing, and we'll get going in
It
here.
should
I think
just-
we're having a cool discussion here.
Yep. Okay. Bye everybody for a minute.